Okay, so this is cell reproduction. We're going to talk about both sexual and asexual reproduction, but today we're just going to focus on mitosis. So first thing we need to cover is, why do cells reproduce? So why do cells need to reproduce?
Maybe like repair damages.
Repair damages. If you get cut, obviously you wouldn't heal if your cells could not reproduce. Good. Do you have the same number of cells you had when you were born? So what's another reason?
Growing.
To grow. Good. And third reason, if none of us reproduced, we would become extinct. So the three reasons why cells reproduce would be, number one, to allow an organism to grow, because organisms do grow and develop. So we obviously are made of more cells now than when we were born. Number two, to replace damaged cells. Cells tend to age. They can become damaged. Some cells only last a short period of time. Skin cells, for example, only live 35 days. You get a new skin cell, it works its way to the surface. By the time it gets to the surface, it's dead. It's sort of like this waterproofing packet of protein, and then it flakes off and becomes the dust in your house. So if you couldn't reproduce your skin cells, well, you'd have a lot less dust in your house. But you'd also have no skin after about 35 days, because your skin cells don't last that long.
So you'd just be like flesh.
I guess you would just be like, yeah, you would, yeah. If you ever had like a broken bone and they put it in a cast, when they take the cast off, it's all crusty and nasty because basically the skin cells couldn't flake off, so it just builds up. Yeah, I don't know. I've never had a broken bone because I don't really do anything. Yeah. All right, and then of course to make more organisms. So the only thing that really gets passed along when cells divide, or the main thing that gets passed along, is the genetic information. That's the most important thing, because that's all the directions for building the organism. So our genetic information is called our genome. Like the Human Genome Project, which was basically something that the CDC had scientists who basically analyzed the entire DNA, 46 chromosomes worth, of several humans, and they were basically able to map it out. And then from there, we've now learned, for example, where certain genes are located. Oh, on chromosome 6, there's this little section, and if your section has this particular code, then you are going to have Huntington's disease, or you have an 80% chance of breast cancer, or whatever. So we've been able to map out the location of a lot of genes that code for different information. Unfortunately, a lot of times, some of these genes, we know where it is, and we can tell you if you have it, but then there's nothing that you can do about it yet. We can't fix it. So that's where the future, lies. That hopefully, not only in the future will we be able to say, yes, you have that breast cancer gene, but that we'll be able to say, and we can go in and fix it and make sure that you then don't, go on to develop breast cancer. So that, that would be what our hope would be for the future. Okay, we're going to focus on eukaryotes because we're made of eukaryotic cells. And I know that you've learned this before. The DNA is in the nucleus, and you cannot actually see the chromosomes most of the time. The DNA is in this mass called chromatin or chromatin. So if you were to look at the nucleus of a non-dividing cell, what you'd see is, if this was our cell, and then this was our nucleus, you'd see a dark area. What's that? Do you remember? The nucleolus. Good. And then you would see the darker gray stuff, and it would look just kind of like this, and that's the chromatin. And that's basically your DNA and protein kind of combined, and you really couldn't actually see the chromosomes themselves. Now the reason behind that, well, before I say that, just a quick comment. Because it's 60% protein, in the early discoveries, 1920s, 1930s, when they were first learning about our heredity, when they did an analysis of what was in the nucleus, they knew that something in the nucleus was passed on to offspring. Because they understood that. They could see, okay, sperm, egg, nucleus, they fuse. So something was getting passed on. They actually thought it was the protein, though. Because since it was 60% protein, it's like, well, there's a lot more protein than DNA. Protein must be what's important. Later, we discovered that was incorrect. It's the DNA that actually gets passed along. The protein is actually very important though, and I'll show you this on the next slide. It stabilizes everything. So the DNA is associated with protein. The proteins that DNA is associated with are called histones. And going back to biochemistry, because I know how much you love biochemistry, DNA is made of nucleotides, which are sugar, a phosphate, and a base. The phosphates make the DNA overall have a negative charge. But what happens is the proteins, the histones, have a positive charge. Since opposites attract, the DNA literally coils up around these histones. So it's sort of like if I gave you a pile of string, you didn't want it to get all knotted and whatever, the most logical way to take care of it would be to kind of wrap it around something. So it's sort of the same way you do if you've ever had like an extension cord, or how about earphones, or those things, the cords get all tangled up. But if you wrap them around something, now they're more organized. So that's basically what happens with your DNA. Your DNA is wrapped around these special proteins called histones. So this is what it looks like here. Here's your double helix. And then our double helix, if we were to look here, it's this little stringy stuff, and it's wrapped around these proteins. And then the proteins are all coiled up around each other. And so what a chromosome really is, when we look at a chromosome, is this really coiled up thing. When we draw them, we kind of give the implication that it's what you really have in a chromosome is like one double helix, and then a second double helix, and that's a chromosome. And that's not the case. The amount of information in a chromosome is huge. If you were to uncoil it, it would be this piece of string that would go all the way down the hallway. But it's coiled up in a very organized way, so it doesn't get tangled, and so that it can be unwound in certain areas when the DNA is going to be copied. Because remember, the whole job of DNA is that it's the directions for making proteins, which is the most important thing in our body, really, the proteins, because they are for movement, they are pigments, they are enzymes, they are hormones, they transport things in and out of the cell because they're membrane proteins. So, so the DNA, the code for making proteins, if I want to read a recipe, I have to open up the recipe book. And I flip through and I can get to where the recipe is. Or imagine I have to take pages out to get to the recipe. But if I want to now give you the book, loan the book to you, I want to put all the pages back in order, pack it up neatly so nothing gets lost. And that's what's basically happening with DNA. Chromatin is when the DNA is unwound. And that's when the DNA, like a book, can be read. The directions can be read. When the DNA coils up is when the cell's getting ready to divide. The DNA is not doing anything then. It's like closing the book. But now, when the cell splits, each new cell will get a copy of the DNA in a very nice and neat way. So it coils up only when the cell divides. Here's another diagram of it. You can see here the double helix. And then this little region of double helix would be this, wrapped around these proteins. And that would be just this little part of this super wrapped structure, which would be this. And then that would become just one tiny little fraction of the chromosome. And like I said, since it's like a book and it has to be read, some portions of your chromosomes stay coiled up. They only uncoil at specific times. And we don't completely understand what causes them to coil and uncoil. We know that there's protein factors and RNA factors that turn certain genes on at certain times. For example, you don't go through puberty when you're born. That would be a problem. It would be very awkward. So instead, you have certain triggers when you get to a certain age that part of your DNA turns on. It's not like you have new DNA all of a sudden. All of your cells have the same DNA. But DNA is turned off and on at different times. And in certain types of cells, only certain portions of the DNA are turned on. So in muscle cells, only portions of the DNA are turned on that are involved in what muscle cells do, muscle contraction or whatever. Whereas in white blood cells, different portions of the DNA are turned on. So all of your cells have the same chromosomes, but they uncoil in certain areas to copy just those sections to make whatever hormones, enzymes, or whatever that particular cell needs. So here's another thing about chromosomes. People make the assumption whenever, if I was to say draw a chromosome, I would assume most people would draw an X and maybe put a little circle in the middle of it. But here's the thing. Early in a cell's life, it doesn't look like this. I mean, it's not coiled up either way, but if I was to coil up a chromosome, it would actually look like this. If I was to take a brain cell and somehow get the chromosomes in a brain cell to coil up, the individual chromosomes would look like this, not X's. Because here's the thing, the only reason chromosomes look like an X is because if that cell's going to divide, it needs to make a copy of its information, and that way, when the cell splits, each new cell will get a copy of the identical information. So technically, a chromosome doesn't need to have two sides. This is just a carbon copy of the original side. So if a cell's never going to divide again, like a brain cell or a liver cell, the chromosomes would look like this in a cell like that, because they wouldn't need to make an extra copy because they're not planning on dividing.
So the only time they become X's is when they're going to divide?
Correct. The only, first of all, during the time when the cell's not dividing, cells that are never going to divide never make an extra copy of the DNA. But even so, the only time you would actually be able to see the X's physically is when the cell's dividing. Because if they're all coiled up into X's, then they're not doing anything else. Think of it this way. You go through all your normal things that you go through in your house. You know, you're cooking, you're doing this. If you're going to move, if you've ever moved, nothing is going on normal in your house. Everything is shut down because you're packing stuff into boxes and you're boxing it up in the car. And you can't, you know, cook because you've packed up the plates. And, you know, you can't watch TV because you've packed up the, you know, DVD player or whatever. That's what's happening here. When the cell's dividing, everything else shuts down. Once it splits, now this will unwind. You'll only have half the amount. You'll have this amount. And it'll unwind, and now it can be read, and the cell can go about its normal business. Just like once you're back in your, you know, your new house and everything's back in place, okay, now I can cook again. Now I can watch TV again. Oh, now my favorite soap is back in the shower again, or, you know, whatever it is. So this is showing an electron microscope picture of a chromosome. So the two sides of a chromosome are called chromatids or chromatids. And what holds them together in the middle is called the centromere. So each individual side is not a chromosome, it's called a chromatid because it's part of one chromosome.
Calder, can you please send Luis Ponce to Student Services with his belongings?
Okay.
Thank you.
There's a protein in the center you may not have learned about called the kinetochore. It looks like this. It's spiky. If you remember learning about cell reproduction, because I know that you've learned about it before, you may or may not remember it, the chromosomes would line up in the middle, and then you had these fibers called spindle fibers, whoops, and they would come across and be connected to the chromosomes. I don't know if this looks familiar to you at all. And then the chromosomes split apart. In order for this to happen, the chromosomes need something to stick to these fibers. And that is what this protein does. It's sticky. If you've ever gone through the grass, high grass with socks on, and you get those little stickies that stick to your socks, well, think of this as being like that. This is what lets it attach to the spindle fibers so that it can be pulled apart.
Where is the kinetochore and where's the centromere?
Okay, fine. The centromere is more of a name for this area.
Oh, okay, so it's not an actual thing?
Yeah, it's more of a name for this general area where they're connected to each other. And then the kinetochore is the name of the protein. And I wouldn't ask you a question on a test where like centromere and kinetochore were both choices.
Okay.
So you can, and they're not exactly the same, but they could be interchanged on a test and they would be referring technically to the same thing.
You know how you said that like a chromosome only has one side until they divide? So then would that side still be called a chromosome, or would it be called
No, as a matter of fact, that's a good point. This is called one chromosome, right? But right after this step is when the chromosomes pull apart. If you remember, what happens next is that they now get pulled to opposite sides. So now, this is now called a chromosome now. They are not chromatids anymore. Sort of like before a baby's born, it's a fetus. As soon as it pops out, it's a baby. It's not, you know, you don't refer to it, oh, look at my new fetus. You know, it's a baby. So it's kind of the same thing. These are chromatids as long as they're attached. This is one chromosome with two chromatids. This is anaphase, when the chromosomes divide, these are now called chromosomes, not chromatids anymore.
So each individual strand is a chromosome?
Yes, but when they're connected, they're chromatids.
Okay, got it.
All right. So this is just another picture. This is showing again, this is one chromosome. If this cell is going to divide, it'll go through a stage where it'll make a copy. And then when the cell actually divides, each new cell will get a copy of that chromosome, which makes these two daughter cells, clones of the